Good morning, students. Welcome to the biology class. Today we are going to start with our new chapter, cell cycle and cell division. It is the part of your unit, cell, a basic unit of life. In this chapter, we will discuss that how the division of the cell takes place and how the new daughter cells are being formed and how the life is going to be processed. Cell cycle is the first part of this chapter. Introduction. All multicellular organisms start life as a single cell, the fertilized egg or zygote. So after the fertilization, when the male and the female gamete, they fused with each other, they form a first diploid cell, that is zygote. Zygote grow by addition of new cells. The new cells arise by division of pre-existing cell. The continuity of the life depends upon the cell reproduction or cell division. Each new cell must inherit not only a nucleus containing the entire genetic information, but also the cytoplasmic organelles required to express the genetic information. So, but also he nahi hai, this is all these events are genetically controlled. In this image you can see that is your parent cell, that is diploid, which is formed after the zygote. It undergoes the division to form the two daughter cells, and from two daughter cells the four daughter cells are being formed and the life continue. So this is the basic concept of cell division or the cell reproduction. What is cell cycle? Cell cycle was described by the Howard and Pillay in 1953. Definition of cell cycle is the sequence of events by which a cell duplicates its genome. Genome means that is the number of the DNA, that is the genome, the genetic material, and synthesizes other cell content, that is your cytoplasm, RNA, protein, cell organelles, and eventually divides into two daughter cells. So what is cell cycle? That is a sequence of events by which a cell duplicates its genome, that is the genetic material, that is the DNA, and synthesize the other cell content and eventually divide into the two daughter cells. In this image you can see this is your NCERT image which shows the cell cycle. It is divided into two phases, that is your interphase, and the second one is your mitosis. Interphase is divided into three parts, that is G1 phase, S phase and G2 phase, and then it comes out with the mitosis or the M phase, which is divided into the karyokinesis and your cytokinesis. So the interphase is being known as the resting phase, or you can say it is a preparatory phase when the cell prepares itself for the cell division, and actual cell division takes place in M phase, that is your mitosis. So generation time. Duration of cell cycle, that is the period between the two successive cell division, is called generation time. It depends on the type of cell and external factors such as temperature, oxygen and food supplies. Bacterial cell divides every 20 minutes. Epithelial cell of intestinal lining every 8 to 10 hours. And onion root tip cells may take 20 hours. So that variable. Mammalian, that is human, cell divide once in approximate every 24 hours, and yeast cell only in about 90 minutes. So these are the generation times for one division to the next division. Phases of cell cycle. The cell cycle consists of two basic phases in somatic cell. In our body, we are having two types of cell. One is somatic cell, another is reproductive cell. The cell cycle, the M phase, takes place in the somatic cell, which helps in the growth and the development of the body. And that is your interphase and M phase, or mitosis, or you can say the meiotic phase. So first we are going to discuss about the interphase. Interphase represent the phase between the two successive M phase. Interphase is most active stage in the cell cycle when the cell prepares for the division phase. It is the resting stage of the cell and occupies the longest duration, approximately 95% of the duration of cell cycles. Suppose it takes 24 hours. So only 1 hour is for the M phase, in which the actual division takes place, and the rest 23 hours are for the preparing the phase, that is called as an interphase. It is divided into three phases on the basis of biochemical studies, that is gap phase 1 or G1 phase, S phase or synthetic phase. So this is your synthetic phase, gap phase 2 or G2 phase. So first we are taking the gap phase 1 or G1 phase. The cell prepares itself for the duplication of chromosomes. The cell is metabolic active and continuously grow, but its DNA is not replicated. Replicated means multiply. It is not going to be doubled. RNA, protein and nucleotides are synthesized and accumulated. Energy-rich compounds are formed. Volume of the nucleus and the cell increase. So basically, the G1 phase is known as the growth phase, in which the volume of the cell is going to be increased. Next is your synthetic period or S phase. This is very important. It is a period during which DNA replication takes place and the amount of nucleic acid, means the DNA, doubles. That is 2C to 4C. There is no increase in the chromosome number. If the cell is having 46 chromosome, number of chromosome remains the same, but amount of the DNA is being doubled, which remain the same. That is, if 1N haploid or 2N, so they will remain 1N or 2N. Centriole in the animal cells initiates their duplication in the cytoplasm, because they also play the very important role during the division of cell. Gap phase 2 or G2 phase, cell growth continues and RNA, new proteins are only synthesized. Density of nucleus increases. Macromolecules are synthesized for cell organelles. Spindle formation and cell growth takes place. So these are the three phases of your interphase in which the various events takes place. Number one is your gap 1, in which the growth of the cell, volume of the cell, that increases. The organelles, they becomes double in the size, but no replication of DNA takes place. Whereas in the S phase or in the synthetic phase, the replication of DNA takes place, that becomes doubles the amount from 2C to 4C. But the chromosome remains the same. So you have to be very careful about that particular point, that DNA doubles but the chromosomes remains the same. Okay, the next in gap phase 2, the growth of the cell continue, RNA, proteins, they are synthesized and the density of the nucleus increases, spindle formation, cell growth, they start synthesizing. The next is your M phase or the meiotic phase. This phase represents the phase when the actual cell division or the mitosis occur. It is the phase when the replicated DNA is distributed to the daughter nuclei in complicated series of the cell. And it involves two stages. Karyokinesis means the process of division of the nucleus, and cytokinesis, it is the process of division of cytoplasm, and then the two daughter cells are being formed. So this all events comes under the definition of cell cycle, in which we said that is a sequential events in which the cell duplicates and the cell divides into the two daughter cells. So by this the cell will ultimately divide into the two daughter cells having the same number of the chromosome as well as the DNA. So in this image you can see that is the interphase, that is your G1 in which the growth takes place, S in which along with the growth the DNA replication takes place, and this is G2 and growth and the final preparation of the division. And then you are having the mitosis or the M phase, that is your karyokinesis in which the nuclei is going to divide and they are having the prophase, metaphase, anaphase, telophase, and little at the end you are having a cytokinesis by which the two daughter cells are being formed. Now next there is one more phase that is called as the quiescent phase or G0, means when the cell that stop dividing. So it is a stage where the cell do not divide further and they exit G1 phase and enter into an inactive stage of the cell cycle. Cell in this stage remains metabolically active but no longer proliferate, means multiply nahi karega, unless called on to do so depending on the requirement of the organism. And these include nerve cell, skeleton muscle cells, mature circulating WBCs and RBCs. So you must have seen कि जब कभी चोट लगती है तो उसके बाद दोबारा से स्किन पे रिकवरी हो जाती है. The wound is going to be healed. This is because now the cell which is in the G0 stage, means that is in the inactive stage, they start multiply. They helps in recovering your wound and forming a new skin, and after that they will again go back to the inactive state. That is your G0 phase or the quiescent phase. Means they are not dividing. They will divide only when they are being asked to do so, unless they are metabolically active but not dividing. So that phase is known as G0 or the quiescent phase. So with this we finish our cell cycle. This is very important question. Along the diagram the question may be there, that is describe the various events of the cell cycle. So you have to describe about the interphase and then the three phases of interphase, that is G1, S and G2 and M phase. G0 is a separate question. That is what you mean by the G0 phase, that is your NCERT question 2. And sometimes a question comes ki resting stage. We call the interphase as a resting stage. Is it true? Justify. So we can say the interphase is the resting stage, but it is highly active stage. It is misnomer because the cell is most active during the interphase because it is preparing for the actual division. So it is not the resting stage. Basically, it is a preparatory stage when the cell is preparing itself for the division of the cell. So sometimes the question is there whether it is correct to justify the statement like this way. You have to copy all this particular content in your biology notebook along with the diagram. They are very, very important. So our next lesson will be on the mitosis, which is being known as the equational division, in which we discuss about the karyokinesis and cytokinesis. Till then, stay home, stay healthy, stay happy. Thank you.